research work on predictive microbial risk in urban systems. The urban environment are not isolated. They form an interconnected microbial networks. Microorganisms move from one environment to another, for example, from the sewage system to hospitals, ambulances, and to public transport. Despite this, we still cannot predict how microbial risk actually happens. So in this, what we know is this environment they mostly mediate pathogen transfer and also antimicrobial resistant movement, but yet their ecological organization and their resilience is not yet quantified. As I mentioned earlier, this environment form an interconnected microbial networks, but networks where contamination potential and contamination potential really defines biosurveillance. But yet we cannot measure the system behavior. We don't know which environment dominates. We don't know which are vulnerable, and we don't know when contamination becomes meaningful. They have so many of these studies. This is because we have a lot of studies on these built environments, but most of them, they are in isolation. For example, we have the studies from the Metasub Consortium that focus on the urban on the transport systems. We have a study by 2017 that focused more on ambulances from the United States by O'Hara, and so on. So the thing is, the problem is, despite we knowing all these things, we cannot, because of the studies are in isolation, we cannot do all these things. We can't measure the system behavior. We don't know which of the environment dominate. We don't know which ones are vulnerable, and we don't even know when contamination becomes meaningful. So it is important for us to understand these dynamics. We need a kind of genome-resolved frameworks that are capable of linking this community composition with the ecological order. So the question is, can we really predict microbial risk in these microbial systems? The first thing we did is we collected 767 metagenomes from NCBI SRA the sequence retrieval archive, and these metagenomes, they represent their shotgun metagenomes, mostly the Illumina pair end reads, and from representing 4 of these environments, and these sequences were undergoes processing, and after processing, we also conduct some quality control and pre-processing to have only the high quality decontaminated paired reads, and this is what we use for the downstream analysis. After this, we subject these sequences, the high quality decontaminated reads, to assembly, where we also assembly to generate the contigs, and we only focus with contigs that are having lengths greater than 1.5 kilo base pairs. After we do this, we then we check the quality of these contigs, and then we also do genome binning. And in the genome binning, we use we bin the genomes to generate catalogs, the marks. After we got the marks, we then conduct the replication to focus only on representative genomes across the environment. And after we do this, we also do the taxonomic classification of these representative genomes, and also we do some functional annotation using the RGI and the VFDB, because we are only trying to focus on pathogenic species. So we do our analysis focusing on only the species that are carrying at least 1 or more ARGs, that are only carrying at least 1 or more VFDB or both. So at the end, we collected a subset of our species that are functionally related to pathogenicity, and that is what forms our species profiles. And we use this. After we got the species profile, we do some CLR transformation, and after the CLR transformation, we also conducted batch correction, because we collect these samples from different studies, different environment. We do the batch correction, and we remove the batch, and we use the data, the CLR transport batch corrected data, for machine learning analysis, in order for us to classify the environmental origin, to identify key microbial indicators, and also evaluate the ecological predictability. And after this, when the machine learning works, then we move forward to do a kind of simulation to studies in order to explore how these microbial communities respond to compositional perturbations and to quantify the cross-environmental vulnerability. And after that, we formed a kind of risk scoring, because the final thing is, how can we really form a kind of risk score to really tell the environment that are at risk this thing. But before we start our modeling, we did a lot of analysis. But what we do is we ask ourselves a simple question: that do microbial communities actually differs across environment? And we do a kind of beta diversity analysis. We do a lot of the alpha diversity, and we saw a kind of clear environmental specific clustering with distinct microbial signatures across the environment. Of course, we saw that among the environment, the hospital sewage is highly dissimilar compared to those environment. And when we do pairwise comparison across all of them, the environment are really different, dissimilar, which gives us a kind of a strong signal for us to continue with the prediction. And when we go to the prediction, we go to the machine learning analysis, as I mentioned, in order for us to be able to classify the environmental origin. We use different models. At least 5 models were tested, and we use them using 10-fold cross-validation. And after we have those models, we test them, their accuracy, and then we find the 2 best models that were having this thing, the random forest and the decision tree. These 2 models, we moved together, we continue with them, where after we continued with these models, we subject them to nested cross-validation. And after we conducted the cross-validation with them, we find out the random forest to be highly reproducible and generalizable, and then we use it finally on hold-out data to find the final accuracy. And we saw that the machine learning actually predicts the environment from the microbial composition with about 97% accuracy. And after we do that, we try to check for the future importance. And for this future importance, we use 3 strategies. We first use the Guinea impurity, we use the DT split base relevance, and also finally we use the permutation importance. After that, we rank and we find the best species that are features aggregated across 250 model rows, and we identify the stable microbial predictors. Amongst this, we have the 5 are the down one. We have the Acinetobacter baumannii, we have the Acinetobacter nosocomalis, and we have Bacillus, and so on. These are the down one. They are the ones that are really used to predict the environmental condition. And after we got this, we moved forward for our microbial simulation analysis. And in this microbial simulation, what we do is we define a fraction which we call alpha, and we start with alpha 0 and we have alpha 1. What we mean here is, for 0, it means the environment is kind of recipient, and 1 is like the environment is a kind of like donor. What we mean is we keep adding fractions of this donor environment. For example, when we look at the hospital sewage, we keep adding we keep adding the remaining 4 environment in a kind of fraction. And what we are trying to see is we want to find out a fraction at which the classification, because what we wanted to do is the machine learning we choose the random forest in order for us to be able to, since it can be able to discriminate this environment at almost 97% accuracy. What we are trying to do is we want to see if we have this environment and we are introducing other environments into it at a kind of contamination level, how much, at what point is the classification changing? And that's what we do. That's why I said the 0, and then the normal donor environment we're adding is at 1. So we can see, for example, in the sewage, in the hospital sewage, when it is the recipient and we keep adding, for example, hospital environment, we can see at very little fraction, the classification of the hospital sewage started changing, and we define a kind of probability at 90. What is the smallest fraction that this changes at 80% probability? And we can see that when we compare the hospital environment and the public transport and ambulances, we see that the hospital sewage classification changes at like 0, lesser alpha value compared to the distance. So it means with little amount of hospital environment, adding hospital environment, the classification of hospital sewage can change. But when we consider the ambulances, we see it can resist, the hospital sewage can resist until we go above 0.7. Similarly, when we looked at the other environment, we have the same cases. For the hospital's environment, we saw that it can stay for more. We need more contamination of this environment before you change it. When we see the ambulances, we see with very little fractions, the classification changes at 80% probability. And when we look at public transport, we can see it is kind of intermediate behavior. So in short, we can say that the sewage behavior depends on context and that it's sensitive to some donors, for example, the hospital environment, but not to others. And meaning they are not universally dominant despite high diversity. As I mentioned, we saw high diversity in the beta diversity. And when we move to the hospital microbiomes, we saw that it is most compositionally stable and that we can see they resist under high levels and that they maintain core microbial, meaning they maintain their core microbial identity. And when we go to the ambulance environment, these are the highly vulnerable environment which are they shift rapidly at very low contamination and they are easily overridden by other environments. While the public transport, as I mentioned, they form a kind of intermediate behavior, moderate resistance to contamination, as we can see, and they act as a kind of bridge between the other environments. So after this at the 80%, I said, is what we call the MDC, minimum detectable contamination. And then to quantify this dynamics, we use the MDC matrix. And this MDC matrix, lower values means a kind of stronger donor influence. For example, when we see the hospital environment, they act as a kind of, because they are very low values, they act as the strongest donor, overriding others at lower contamination levels. It means you require very low amount of it to override the other environment. And when we see the public transport also shows strong but targeted effect, especially on ambulances. But when we talked about the recipient, we can see that ambulances are most vulnerable, and then while hospital and sewage systems are the most stable. What we found here in general is that the microbial influence is normally asymmetric, with some environment becoming dominant and some other environment as vulnerable. So we also try to use a concept of entropy here to really define a tipping point. What we mean by tipping point is, for example, when we see the hospital environment and ambulances, we try to also, as in an increment, 10% increment, 0% is the normal environment. And when we keep adding the contaminated environment or the donor environment, when we keep adding 10% of it, we try to check also here with entropy to see at what point do we this thing. So from the beginning, we can see that the entropy is low value when we have the normal environment. And then as we started adding, the entropy started increasing until we reach a point where the entropy reaches the highest level and then comes back. That point where that happens, we call it the tipping point. And that point normally we can see is different between environment. For example, when we are using ambulance as the recipient and we are adding small fractions of the hospital environment, we have a tipping point, early tipping point is established when we compared with the hospital environment and the public transport. Similarly, the public transport and ambulances also, we saw the tipping point established early, trying to tell us that the ambulances also is a kind of more fragile environment compared to the others. So this tipping point is normally where the identity becomes ambiguous, and after that, the system goes back to maybe change the model tries to change the classification to the donor environmental condition. So at the end, to summarize these dynamics, we integrate the multiple metrics to quantify the contamination risk. We used three, but to make it more robust, we try to follow this at three. We use three weighing skills. We have the primary weighing skill, where we use the primary weighing skill, we have the equal weighing scheme, and we have the PCA-based loadings. And in all this, we use the overriding frequency, we use the entropy, we use the richness and the variance. This, we pull them together for the primary. We give the overriding frequency 0.4, we give the entropy 0.3, and then richness 1.5, 1.5. And then for the equal weighing, we all give them equal weighing to really see how will we get the risk environment. And from our result, the public transport and the sewage represent the higher risk environment, while the hospital the public transport and the sewage represent the high risk environment based on this, while the hospital environment are more stable and ambulances, although vulnerable, they show lower overall risk. These patterns of dominance and vulnerability are what we summarize here as the risk score. Then after this, we try to really combine this with the WHO priority pathogens, and we tried to do this, and we found out that the WHO priority pathogens were almost detected across our environment, and then which meaning that these WHO priority pathogens, they are not random. Instead, they are structured within the microbial communities. What it is important is not just that these pathogens are present, but they occupy central position in the network linking environments. So from this, our key findings shows that the urban microbiomes, they form a kind of connected systems, and the microbial identity shifts at contamination thresholds, and that we find out that environmental influence is asymmetric, showing a kind of sourcing dynamics, and that the contamination risk varies across environments. We published this as application notes called the GRUMB framework, and also we have the companion article, which we show the result here. These are some key terms that were developed from the work. We like the contamination simulation, recipient environment